Today we are going to start our experiment number 5, that is chromosomal DNA isolation from plant tissues. Okay, and I will elaborate it. What's the method? What's the strategy behind that? And then we will go to the lab and we will do it practically. Okay, the first thing that we must notice is that the plant tissues have a very rigid cell wall. Okay, and we can break it through various means. If we have protoplasts or the cell culture, then our approach might be that we can add some cellulases to dissolve the cell wall and then some detergent to dissolve the membrane. But in our case, we usually isolate the DNA from plant tissues directly, okay? And therefore, we use mechanical method of cell wall disruption. So the method that I'm going to discuss is very commonly known as CTAB method for DNA isolation. Okay, so what we will do in this case is we will isolate or we will harvest some tissues like leaf tissues. What we need for this experiment is we need at least 100 milligram of tissues, and these are usually leaves. Okay, and we grind these tissues in liquid nitrogen, usually to stop the nucleases from degrading the chromosomal DNA. Okay, but if we go very quickly through the process, sometime if we don't have liquid nitrogen available, then we do proceed actually without liquid nitrogen very, very quick, okay, to avoid any degradation of the DNA. So we will try, I'll see if we have a liquid nitrogen. I hope it is available. And we will grind those tissues in liquid nitrogen. And prior to that, we will chill the pestle and mortar with liquid nitrogen, and the temperature of liquid nitrogen is minus 190 degrees centigrade. Okay, so we will chill that apparatus first, and then we will put our tissues in the pestle and mortar, and we will grind to obtain the powder. That's a fine powder which will indicate that all the cellular structures like the cell wall and the nuclei, they have been broken down and the DNA has been released. Okay, so this is the second step. The third one would be that we will suspend this powder in a mixture of 2-CTAB style trimethylmonium bromide. This compound is actually, it selectively precipitates the polysaccharides and the proteins, and DNA does not dissolve into it. Okay, so we will add 2-CTAB to about 100 milligram of the tissues, ground tissues in the powder form, and the volume of this solution is about 700 microliters per 100 milligram tissues. Okay. After resuspending this solution, what we do is that we can go directly to heating the solution to about 65 degrees centigrade for 30 to 45 minutes. And that time is enough actually to remove the DNA from the other cellular materials like the cellular debris, the polysaccharides, and the proteins. So after the treatment at 65 degrees centigrade for 30 to 45 minutes, what we will do is that we will take the tube out to the room temperature, okay, and then we will add chloroform isoamyl alcohol. Now this is a protein denaturing agent. There are several protein denaturing agents which are used in the laboratory. The first one is the phenol, the concentrated phenol, and that is used when high amounts of proteins are present. Then we can also use a mixture of phenol and chloroform. In this case, whenever I say chloroform, it means that it is 24:1 ratio of chloroform to isoamyl alcohol. Isoamyl alcohol is actually the suppressant of bubble formation, okay? So in order to avoid the bubble formations, we add 1 part of isoamyl alcohol to 24 parts of chloroform, okay? And the protein solutions, they usually, if you mix the protein solution, they form a lot of bubbles, air bubbles. To avoid that, we can use isoamyl alcohol along with the chloroform. And chloroform is actually the weakest protein denaturing agents. So after heating at 65 degrees centigrade for 30 to 45 minutes, we believe that most of the protein, they are gone, but there are still some remnants of the protein. So what we do is, our next step is that we have actually a mixture of cellular debris and the entangled polysaccharides and the proteins. So what we do is that we spin the whole tube after heating and adding chloroform isoamyl alcohol at about 10,000 to 12,000 RPM for about 10 to 15 minutes. Okay, so what it will do is that it will remove most of the cell debris, and you will get the debris settled along the tube at the bottom of the tube, and you will get a supernatant in the tube after this spin. What you do is that you take out the supernatant into a fresh tube, okay, and then you add equal volume of chloroform isoamyl alcohol. And now you have to mix it. And mixing this solution will further precipitate the remaining proteins which were present here. Okay? Traces of proteins, that is protein traces. Okay? And this treatment, you will gently mix them to avoid the breakage of the double-stranded DNA. Okay? After doing this, you go again to, this is now step number 6. Okay, this was step number 5. Oh, sorry, this is step number 6. And now we will go for step number 7. That is again spinning at 10,000 to 12,000 RPM. What you will get is you will get a layer of chloroform isoamyl alcohol and the other layer of your DNA solution. And you need to precipitate the DNA now directly from that solution. So what you can do is you can take out the liquid into a new tube. Okay, don't touch chloroform isoamyl alcohol layer. Okay, just take the supernatant into a fresh tube, and to that tube add 0.6 volumes of isopropanol. So that's it. Mix the supernatant with isopropanol and keep that tube at minus 20 degrees centigrade for 30 minutes to an hour. Okay, and this will precipitate the whole chromosomal DNA. At the end of low temperature incubation, what you do is that you go for a spin again at 10,000 RPM for 5 to 10 minutes, and you get the DNA pellet at the bottom. You can wash the DNA pellet with 70% ethanol, and then you can dissolve it into the appropriate volume of water, and that is dependent on the size of the pellet. So this is the small description of the experiment that we will be doing for the isolation of chromosomal DNA. And now we will go to the lab to do it practically, and I will try to show you each of the steps that we have discussed here on the board. Thank you.
Okay, now we are in the lab, and here what we have got is we have the leaf tissues which were stored at minus 80 degrees centigrade. We have taken them out, and we have cut some pieces out of these leaves, and what we will do is that we will put the liquid nitrogen into this pestle and mortar to chill it down, and then we will grind these tissues. The liquid nitrogen is available here in the flask, and then we have some other stuff that we already saw, that we have some CTAB solution, some Falcon tubes, some test stand that we will be utilizing along with the other important reagents that we will use here in our experiment. Okay, now we're starting the grinding process. So first of all, we will pour some liquid nitrogen into this pestle and mortar so that we can cool it down to minus 196 degrees centigrade. So she is immersing now the leaf tissues into this pestle and mortar, and the grinding process has been initiated. So it will take a little while until everything is ground to the powder form, and you can see that the tissues are breaking down into smaller pieces, and as the liquid nitrogen evaporates, it will be easier to grind these tissues into the powder form. Okay? Yeah, it is almost very close to the powder form, and a little more grinding is required actually to get a very fine powder out of these tissues. Do we need to add some more nitrogen, or it's okay?
I guess it's fine.
Okay. Okay, she has, crushed these tissues enough, and they are actually in a bit rough powder form, but still we can isolate the DNA from there. She will now take these tissues into the Pandolf tube. So Aruch, you can go ahead. So what she is doing is just picking up the tissues into the tube. So she already weighed the 100 milligram tissues, which she started with the grinding process, and this way she is picking up the tissues from the bottom of this pestle into the Appendov tube so that we can initiate the extraction of DNA by adding CTAB to it. She's starting now with the addition of CTAB, and if you remember, she will add about 700 microliter of 2x CTAB into these tissues. And what we are going to do then is that she's taking up the CTAB solution. And the solution is being added to the Appendov tube containing the ground tissues. So she can mix them with 1 ml pipette pen very gently, okay, because they are the powder form, so they will not actually clog the tip. They can easily pass through, and you can then gently mix them like this. This is the hot plate that has been set to 65 degrees centigrade, plus minus 1 degree centigrade is okay. And she has now placed this tube in this hot plate, and the tube will remain there for about 30 to 45 minutes until the digestion of these broken tissues has completely occurred and the DNA has been extracted from the tissues. Okay, we will take out the tube from here now, and we will now spin this tube. You can see the most of the material over time has been settled down, and the extraction of the cellular contents can be visible, and you can see the green color of the chlorophyll, which has also been extracted from these leaf tissues at high temperature. What we have done is that now we are starting the spinning of this tube that we placed at 65 degrees centigrade to remove the debris, and this will be done at the full RPM, as I mentioned, from 10,000 to 12,000, but we can go to 13.3 thousand because this machine is capable of doing that. Okay, and we will do it for 10 minutes, and then at the end of the spin, I will show you that how the debris has been separated and how the supernatant looks like. The machine has been stopped, and we will take out these tubes out of the rotor, and let's see how the pellet looks like. You can see that the pellet is very much clear and settled down in the bottom of the tube, and the supernatant is enough and clear for pipping out into a new tube. Now we are taking out the supernatant from the Appendov, and we will transfer it to a fresh tube so that we can treat this supernatant with chloroform isoamyl alcohol to remove the traces of proteins from this mixture of the DNA and the cellular contents, free of cellular debris now. Most of the proteins and polysaccharides, they have been removed by the CTAB, okay, and the remaining traces of proteins will be removed with the isoamyl alcohol chloroform. So how much volume you have taken? It is about 600 microliter that she was able to pick from the spin tube. Okay, so this is chloroform isoamyl alcohol that she will be using, and remember, chloroform isoamyl alcohol is always 24:1 ratio. Okay, that CIA on the label indicates chloroform isomyl alcohol, and this is how the tube appears after you have added the chloroform isomyl alcohol to this tube. So where is the supernatant that we took the tube? It is on the top. Okay, the chloroform isomyl alcohol settles down at the bottle, but we have to mix it actually, because chloroform isomyl alcohol is a protein denaturing agent, and we need to remove the proteins. So we will gently mix it so that all the proteins get trapped into this chloroform isomyl. You can see that the turbidity of chloroform isomyl alcohol has increased, which means it is extracting out some fractions of impurities from the supernatant that is having chromosomal DNA in it. Can you mix it a little bit more? Yes. So the top of the supernatant is more clear now. You need to mix it more. Let's see how much clarity we can get from this tube. So we are removing actually the chlorophyll from the supernatant tube. Okay? The more chlorophyll is removed, the more clear would be our supernatant after spinning. Okay, I think it's very well done. You can see a middle layer here, and that middle layer is showing whitish specks or whitish precipitate. That is actually the protein that has been removed from the supernatant where the DNA is present. Okay, so we will spin this tube at full RPM just like before, at 13,300 RPM, and we will collect the supernatant one more time, and again we will be spinning this tube at 13,000 RPM for about 10 minutes so that we should get a very clear supernatant. So we will come back when the machine stops. The machine has stopped, and we will take out the tubes from this rotor. And let's see how it looks like. Yes, I can see actually, Aroj, this whitish, let me focus it. Yes, we can see the whitish pellet at the meniscus of the two layers. These are all proteins which have been separated. All the chlorophyll content has gone down to the isomyl alcohol, and the supernatant is very much clear. And this is the one from where we will precipitate now the chromosomal DNA by adding 0.6 volume of isopropanol. So you can see that this extraction has been quite good. You can clearly see the white protein layer in here. Okay, and we need to separate the supernatant very carefully without disturbing this layer. Aruj, I would recommend that take about 450 microliter of the supernatant into a new tube. Okay, we are taking this supernatant out by adjusting the pipette man to 450 microliter, and meanwhile you can calculate the 0.6 volume of 450 microliter. That is the volume of isopropanol that will be added to the supernatant to precipitate the chromosomal DNA. Okay, so we have calculated it already. You can do it yourself. Okay, so the supernatant is collected in a new tube. You can see it. And now we will add isopropanol to this one. So what is your calculation? What is the 0.6 volume of 450 microliter. Arush, did you calculate?
270 microliters.
That is 270 microliter. So, and this is what she is doing. She's adding the isopropanol into this tube, and it has been added. Do you see any whitish precipitate at the point, at the moment? Can you mix it gently? Very gently. Okay. And we have mixed that all. You cannot see the DNA with the naked eyes at the moment, but what we will do is that we will keep this tube at minus 20 degrees centigrade for about 30 minutes, and then we will give a spin again, and we will see if there is a white pellet at the bottom, and that white pellet would be the chromosomal DNA. Okay, that is the total genomic DNA. Actually, it will contain the DNA from chloroplast and mitochondria as well. So we said total genomic DNA. Usually we put this tube at minus 20, that is the temperature of a freezer. So she will hold this tube here for about 30 minutes. Okay, close the door, and we will come back in about 30 minutes. The tube is out of freezer and it's ready for spinning at 10,000 or greater RPM. And you can see that the material is more whitish, which is indicating the presence of now well precipitated DNA in it. And we will place it in the centrifuge. And we will spin it at 10,000 RPM from 5 to 10 minutes. So it's set for 10 minutes. So we will wait for some time until the machine stops, and then we will have a look at the pellet, at how big is the pellet and how much volume we should use to dissolve it. Stopped, and we will take out this tube. Can you see the pellet? Yes, it's very much clear, and it's quite big one. It's chromosomal DNA from 100 milligram tissues, and you can see a big white pellet in there. And what we will do is that we will decant every liquid which is inside the tube, and we will wash this pellet with ethanol to remove all the remaining salts which are associated with DNA. She is decanting the liquid into a beaker, and she will remove the last traces of drops by putting this tube inverted on a tissue paper so that all the liquid is drained completely. And Aruj, you can wash this pellet now. Can I see it again? I mean, in the absence of the liquid. Yeah, it's quite clear, but can you give it a contrast? Yes, it's very clearly seen. It's very clear. Okay, so go ahead and give it a wash with 70% ethanol.
200 microliters.
That's 200 microliter is enough actually for this washing. Okay, and then just mix it a little bit and give it a spin again at the same 10,000 RPM. Just for a minute is fine. The machine is stopped and we will take up the washed pellet. Now again, drain this ethanol completely. Just tap gently, yes. So we need to dry this pellet. We need to evaporate the ethanol before we put some volume of ethanol into it. And I think we can dissolve this pellet. This is a big one, 50 to 100 microliter, whatever you want. So even 100 microliter is okay. So this is a big pellet actually, okay? So you know what happens most oftenly in the experiments when we do it with students in the lab, because they are very much new to these experiments and they don't know that the traces of ethanol, if they are left in the solution of DNA in water, so when you load them onto the gel, in the loading die, everything will pop up. It will not settle down into the well. The reason is the presence of ethanol actually reduces the density of that material which we are piping out into the well. So remember that we need to remove all the traces of ethanol by drying this pellet on the filter paper and then blowing the pellet for a while and smelling it if all the ethanol is gone. Okay, so now the DNA has been dissolved. We are ready to load this DNA onto the gel. For that purpose, we will go to agarose gel electrophoresis and we will prepare the gel first. We have started preparing 1% agarose gel for the analysis of this chromosomal DNA. So we have taken about 50 ml of the TA buffer that is used for the gel electrophoresis purposes, and also it is a buffer that is used for making the agarose gel. On the other hand, we have weighed down 0.5 of agarose that we will add to this 1x TA buffer. Okay, and then we will proceed for microwave. Because this agarose is dissolved at high temperature, it won't be dissolved at the room temperature. You can see that the turbidity is appearing. That is a clear indication that agarose never dissolves at room temperature at all. Please cover the flask with a paper. Okay, the heating is over and we will see that, yeah, we can see it is quite transparent now. The agarose has been completely dissolved and we will cool down this gel to about 50 degrees centigrade and then we will pour it into the gel cassette. We have dissolved the agarose. It is here and we will use this agarose for pouring into the horizontal gel electrophoresis system. And let me introduce you to the components. So the lid has been removed from this apparatus. You can see the combs inside, the gel casting tray. Can you take them out, Aruj? Okay, so we will put the stoppers, which are here at the edges of this gel casting tray. And then we will actually pour the gel after adding ethidium bromide into the gel so that DNA could be visualized after we have run it. So these are rubber stoppers and they have grooves in which the gel casting tray is placed. And after doing this sealing of the caster, this tray is now ready for pouring the agarose gel. We are now starting the addition of ethidium bromide to the molten agarose. and we will use about 2 to 3 microliter of this 10 milligram per ml solution of ethidium bromide that will be added to agarose and will be mixed. This is a highly carcinogenic compound, so be very careful. And now she will put the comb inside this gel casting tray so that the well can be formed within the gel, and you can just pour this liquid into it. So 50 ml is enough for one small gel casting tray, and it will give you approximately 1 centimeter of the gel thickness. So you can see that in this casting tray, the gel is about 1 centimeter thick. Okay, so it will take a little while to solidify, and we will wait until it is solidified, and then we will run it at a voltage that I will tell you that how we calculate that voltage. So let me check this apparatus and show you the electrodes. Okay, if you look at the bottom of this buffer chamber, I hope you can see the steel wire in there. There is one steel wire at the bottom of the black side. Okay, it's clearly visible now. And there is an other one which is close to the chamber which holds the red color terminal. You can see that wire too very clearly here. Okay, yeah. So what you do is that you measure the distance between the electrode at the black terminal and the electrode at the red terminal. The distance between these two wires is measured. And then in order to get 3 volt per centimeter square of the gel, you run this by multiplying the distance between the electrode in centimeters with 3. Okay, that's how you calculate the running voltage for each of the gel electrophoresis apparatus. Distance between the two electrodes you can see is about 16 centimeters on the centimeter scale. Okay, and in order to get 3 volts per centimeter square, we need to multiply this 16 with 3. Okay, that is going to be 48 in total. So 48 volts. If we set that on the power supply, that will give us an optimum resolution of the fragments. The gel is being solidified, and you can see the opacity of the gel, which indicates clearly that the gel is almost solidified, and we are ready to use this gel. Meanwhile, what we can do is that we can prepare our samples. So what we will do is that we will load 2 samples on this gel. One of them is our marker, that is 1 kb DNA ladder, and we prepare this DNA ladder by taking about 7 to 8 microliter of the prepared ladder which is supplied by the company, and we can load that in one of the wells. While for the DNA sample, what we will do is that we will take about 2 microliter of the DNA, and we will pipette it out in a new append tube. We will add 15 microliter. So what you have added, Aruj?
Water and plasmid.
Okay, so she has added 2 microliter of chromosomal DNA preparation that we prepared, and 15 microliter of water to that to increase the volume to 17 microliter. She will add about 3 microliter of the gel loading dye, that is bromophenol blue having xylene cyanol FF, prepared in 10% of sucrose or glycerol to increase the density of this material, so that when we load our sample, it settles down at the bottom of the well very easily. So the DNA sample here is ready now. She's mixing it up. And can I see at the end? Okay, so you can see this is about 20 microliter of the sample which contains 2 microliter of the DNA prep, and we prepare a larger volume because during the loading process, sometimes you can lose some of the material if there is something wrong with the wells. Okay, so 20 microliters cannot be loaded in the well completely. We can take 15 microliter and load that on the gel, and still we save 5 microliter. So that excessive volume is always to be on the safe side, that if something is wrong with the well or with our material drains of the wells, we can use the saved one. Okay, so you can go ahead with the gel. It is almost solidified now. So first she is removing the stoppers, the rubber stoppers from the bottom of the gel caster. Then she will remove the upper caster, and you can see the gel is now solidified. The comb can be seen from within the gel, okay? And if you can also see the thickness of the gel, which is about 1 centimeter. And this is a standard procedure for preparing the agarose gel, that the thickness should be about 1 centimeter, and the well should be well immersed in that gel, so that we can get the wells which are deep enough to accommodate about 15 microliter of the sample. So what she is doing now is she is pouring 1 XTA buffer, and that buffer will be slightly covering this gel. The buffer goes to 2 chambers. One chamber is there on the black electrode side, the other chamber is towards the red electrode side, and both of them, they are connected by the flow of buffer from the top of the gel, okay, so that the circuit for the electric flow can be completed. Now she's ready to remove the comb, and it's always good to pour the buffer before you remove the comb, because it swiftly slides off the gel. Now we will see, actually we will try to see the wells in the gel. Yes, those can be actually seen at the black line. You can put some white paper down there, if you please. Okay, we can see the wells now. We have placed actually a white paper below the gel apparatus, and we have the stopper, red stopper down there. And if we move this, so you can actually, yes, here you can see all the 8 wells very clearly, and we will use these wells for pouring of the samples now. Okay, she is now ready for pouring the material from the DNA sample into the wells, and I will show you the loading. One of the well will be loaded by the marker that I mentioned is 1 kb DNA ladder, and she is pouring one of the wells. So you can see she is quite expert in loading these samples into the wells. The sample has settled down very clearly and neatly within the well. There is no overflow of the sample, and we have everything going down into the well. Meanwhile, she is preparing the second sample, which is the 1 kb DNA ladder. Okay, she will take 6 to 7 microliter of that, which is a pre-prepared, ready to load DNA marker, and she will load it into a second well. Okay, that has also been loaded. So we are using 2 wells here. The one with little material indicates you the 1 kb DNA ladder, and with a lot of dye here, which is of different color. It is actually our own dye, which is chromophore blue and xylene cyanol FF, added to the DNA, and we have 2 loadings. Now we will run this gel at 60 volt actually to complete this running in 30 minutes, and then we will visualize it under the UV transilluminator. It's about 30 minutes that this gel is running. You can see the moisture as usual on the top of the lid, and that's due to the evaporation of water, due to the decomposition of water, due to the flow of electric current. You can see actually the bubbling of hydrogen from this end. The 30 minutes time is over, so you are hearing to the beep. The flow of the current is stopped. What we will do now is that we will remove this lid and we will see how much the gel has run so far in 30 minutes. So you can see here that the dye is away from the wells about 2 or 3 centimeters away, and at the bottom is a lower dye which is bromophol blue, and at the top it is xylene cyanol FF. The quantity of the dye in the marker was very low, so it can be hardly seen here. And we will take this gel now to the UV transilluminator to see what is the concentration or how good is our preparation of chromosomal DNA from these leaves that we collected today from wheat. And we will examine this gel on transilluminator that is placed within the photo documentation system. This is the photo documentation system that we have placed in a small space here in the lab. This is a monitor where we see our gel, and this is the photo documentation system, UVP, and it has got the transilluminator where we place our gel, and when we switch on the UV light, we can actually see the gel from here, okay, and the orange colored bands can be seen here, yes, which are actually representing the ladder and the chromosomal DNA that we loaded onto this gel. Yep, we can see that there. And the same thing we can see from this system onto the monitor. And on this monitor, we can see the RNA which has been degraded and has gone down, while the chromosomal DNA is at the top. It's very close to the well because it is a very high molecular weight DNA, so it remains closer to the well rather than going down to any of the lanes of the marker DNA. So, and we will take the picture out of this. We can email this image to our addresses, and we can then take a record of that. This is how usually we proceed with such kind of images. And that's all about this DNA isolation for today.